Welcome to pointhints.com. Today we are going to discuss about extracellular matrix in biochemistry based on Harpers Biochemistry 31st edition soft copy. Cells are surrounded by ECM component which just connects the cell to the outer substance, so it is called as connective tissue. It protects and provides elasticity in blood vessels, liver and skin. The components of ECM are structural protein, proteoglycan and specialized protein. Structural proteins are collagen, elastin and fibrillin. Proteoglycans, which means glycosamine glycans or mucopolysaccharide plus proteins. Glycosamine glycans, which is nothing but the chain of disaccharide molecule combined with the protein to form a proteoglycan. Specialized proteins are fibronectin and laminin. Defects in ECM. General defect in ECM leads to rheumatoid arthritis and osteoarthritis. Rheumatoid arthritis is an autoimmune disorder in which our own immune cell attack our healthy cells. And osteoarthritis, it is due to degeneration of cartilage. It is seen in aged people. Osteogenesis imperfecta and Ehlers-Danlos syndrome are genetic disorder due to defect in the gene that is responsible for synthesis of collagen protein and other ECM component. Mucopolysaccharidosis is due to defect in lysosomal enzyme in which the glycosamine glycans or mucopolysaccharides just accumulate in our own body. Major components of connective tissue. In mammals, 25% of proteins are collagen. There are 28 types of collagens are present. Component of complement system C1q and surfactant pulmonary protein SPA and SPD are proteins that are not classified as collagen but have a collagen-like domain. They are called as non-collagen collagen. Types of collagen. We are going to look on main type of collagens. Type 1 skin, bone, tendon. Type 2 cartilage. Type 3 blood vessels. Type 4 basement membrane. Type 6 muscle. Type 8 endothelium, which is nothing but blood vessels. Type 25 brain. Type 26 testis, ovary, and type 27 embryonic cartilage and cartilage in adults. Have a look on this order. From outer to inner, first you have skin, then basement membrane, then you have fat, that is connective tissue, then you have muscle, then you have bones. For your easy understanding, collagen molecule have a triple helix structure form, which is nothing but 3 alpha chain combined together to form this triple helix structure. It is a single alpha polypeptide chain which has glycine X, Y, glycine X, Y, etc. It is a amino acid sequence. Glycine is stable because it is a small amino acid which accommodate in limited space. X can be proline and lysine mostly, and Y can be hydroxyproline and hydroxylysine mostly. This residue turn into left-handed helix and form a single polypeptide chain. These chains combine together and form this triple helix structure. In this manner, it turn into right-handed helix and form a superhelix. This lateral area associate together to form this fibril. It is arranged in quarter staggered alignment, which is 10 to 300 nanometer in diameter. These combine together to form a fibers which is 1 to 20 micrometer in diameter, and these fibers associate together to form a large bundles which is 500 micrometer, which is seen in tendons. The content which is explained before is here. The proline is hydroxylated by the enzyme prolyl hydroxylase to form hydroxyproline, and lysine gets hydroxylated to form hydroxylysine in the presence of lysyl hydroxylase by the cofactor ascorbic acid, which is vitamin C, and alpha ketoglutarate cofactor, which catalyze the enzyme activity, are stabilized by covalent cross-link which is present between or within the triple helix unit. These covalent cross links, which is formed by the lysyl oxidase enzyme, which is a copper dependent enzyme, act on the amino acid lysine and hydroxylysine mostly and sometimes histidine. It forms the amino acid into aldehyde by the process of deamination, which stabilizes the covalent bond and important for tensile strength of fibers. Just have a look on this image. This procollagen have some extension. This extension is called propolypeptide extension. It has both amino terminal and carboxy terminal. These are the extensions here. The gene that is responsible for collagen synthesis is COL1A1 gene that synthesizes pre-procollagen enter into the ribosome. It has a signal peptide. It cleaves and tells the chain to enter into the endoplasmic reticulum where the hydroxylation and glycosylation occur. Hydroxylation, which is nothing but proline convert into hydroxyproline by the enzyme prolyl hydroxylase, and lysyl convert into hydroxylysine by the enzyme lysyl hydroxylase in the presence of cofactors vitamin C, ascorbic acid, and alpha ketoglutarate. Glycosylation, which is hydroxylysine plus glutamate, convert to form some substance. The function of the substance is unknown, but without glycosylation, there is no fibril formation. This pre-pro-collagen has a extension which is absent in the mature collagen. That extension has a cysteine residues and it has the amino end and carboxy terminal. In the amino terminal, it has a disulfide bond which is intrachain, and at the carboxy terminal both inter and intrachain disulfide bond is present. This disulfide bond presentation assists the registration of alpha chain to form a triple helix structure. After the formation of triple helix structure, there is no hydroxylation and glycosylation occurs. The above mentioned steps are intracellular. After the triple helix formation with the extension, then it undergo cleavage of the amino and carboxy terminal by the enzymes which is released by the Golgi apparatus. The enzymes are procollagen aminoproteinase which acts on the amino terminal and procollagen carboxypeptidase which acts on the carboxy terminal. After the cleavage, it forms a tropocollagen. This tropocollagen combine together to form a fibril formation in the quarter staggered alignment. This fibril combine together to form a fibers.